Assalamu alaikum wa rahmatullahi wa barakatuh. Good morning, everyone. It's my great pleasure to be invited to this scientific day of the Faculty of Pharmacy at Tanta University. Many thanks to the organizing committee. And my topic today is on exploring new therapeutic targets for colorectal cancer. In this way, we are going to discuss the different vulnerable pathways in which the cancer cell can be attacked through. And in fact, as pharmacologists and pharmacists, we always think of the drug-target relationship. In the cancer cell, there are many targets, there are many vulnerable pathways, and it is essential to explore which one is the most critical that can actually determine the survival of the cancer cell. This is my disclosure. I've received a grant from GSK. However, this fund does not have any influence on the content interpretation or the conclusions presented today. This is the outline of my talk today. I'm going first to discuss several genetic, metabolic, and epigenetic vulnerabilities in colorectal cancer and why they matter. I'll focus on the tumor microenvironment as an important component that determines many outcomes of the cancer and its treatment. I'm going to show several examples from my previous studies, and I'm going to discuss why the advances in experimental platforms has driven the target discovery. I'm going also to highlight a new trajectory in cancer treatment, a little bit paradoxical one. And finally, I'm going to discuss translational changes and future directions. To start with, I'd like to emphasize the significance of colorectal cancer as the third most frequently encountered cancer all over the world and the second regarding mortality in most countries. And this figure shows the difference between males and females, showing a dominance in incidence in males and also in mortality. However, this is not the case in several countries, which means that there are environmental factors that underline the development of the disease. So shifting to the molecular and genetic landscape of colorectal cancer, we will find that the genomic instability pathways are likely to occur in colorectal cancer and is detrimental for the initiation and the progression of the tumor, either in terms of chromosomal instability in form of aneuploidy or copy number variants, or the microsatellite instability, which is usually determined by occurrence of mutations in certain DNA mismatch repair genes, the most famous of which is MLH1 mutations, or it may be rather related to epigenetic alterations, for example, the hypermethylation of the promoter island of the CPG islands of the promoter region of certain tumor suppressor genes. Hypermethylation will lead to silencing of the tumor suppressor genes and finally leading to the development of the tumor. And in fact, from the mutations, we can go to the different molecular subtypes of colorectal cancer. And this is the most, actually, it's not very recent. It was determined since 2015. However, it is a very efficient way of classifying colorectal cancer because the different types of consensus molecular subtypes is well characterized. This means we know which mutations are there, which pathways are activated, and even what prognosis we expect in those patients, and which types of therapies are most likely to be effective. So it comes from the mutation to the molecular subtypes for the sake of serving the patients by choosing the appropriate therapy. And actually, this is a figure prepared by the PhD student, and we published recently in Frontiers. From the mutation also to the therapeutic outcome, because again, I'm emphasizing what I have said in the previous slide, that different mutations that even occurs at different stages of the colorectal cancer, starting from the adenoma to the very initial tumor and then to the advanced tumor, we can actually explore the mutation, and this will tell us which pathways are likely to be activated, and this will lead to the tumor evolution and progression. But even more recently, they found that those mutations have some crosstalks with the tumor microenvironment, so they can determine what type of immune cells can be attracted to the tumor microenvironment, what is the effect on the cytokines in this tumor microenvironment, what is the status of angiogenesis, and of course all those factors play an important role of the response of the tumor to therapy and also the development of drug resistance. So the molecular subtype in colorectal cancer determines the neoantigen load. This means how much the cancer cell is visible to the immune system, even the infiltration of the CD4 and CD8 T lymphocytes, and the representation of HLA-1 and HLA-2 on the cancer cell surface. And also, all of those factors will determine the response of the tumor to the checkpoint inhibition or the immunotherapy. The one that is inversely proportionate or inversely related to all those factors are the bad guys in the tumor microenvironment or the myeloid-derived suppressor cells. Different vulnerabilities may play a role in determining the behavior of the colorectal cancer. So there are clinical and molecular heterogeneities in colorectal cancer. There are also important concepts related to what we call synthetic lethality. And this can be represented shortly in this figure. If we have a normal cell and it has gene A, gene B, and if you inhibit either of gene A or gene B, still the cell can survive. Whereas in the cancer cell, and because it's defective in some particular genes, like here, for example, gene B has a single mutation or is overexpressed, then when you inhibit the interdependent or interlinked gene A, then the cell will die. And this is called synthetic lethality, and it depends on how much we know about those interdependent pathways or synthetic lethality in the cancer, so we can hit the appropriate target. Also, a new concept has developed by targeting cancers with certain molecular background or mutational background. For example, all cancers that are that have microsatellite instability at a high level, they can be susceptible to a particular medication irrespective of their site. So whether we are talking about pancreatic or head and neck cancer or colorectal cancer, all are MSI high, all can be treated by a certain medication. So the key molecular vulnerabilities in colorectal cancer can be based on the oncogenic signaling dependency, on the DNA damage response and replication stress in a particular cancer, on the alteration in the metabolic programs that is also associated with the development of cancer, epigenetic vulnerabilities, and finally, the tumor microenvironment-driven vulnerabilities. I'm going to go through those vulnerabilities in short. So if we go to the first one, going to the first one, which is the oncogenic signaling dependencies, I'll give three examples. The Wnt beta-catenin addiction, which is a very important pathway in colorectal cancer. The MAP kinase pathway that can also be related to KRAS, BRAF, and it's present in many cancers, including the pancreatic cancer. The PI3K, AKT, mTOR pathway vulnerability. And if we go to the first one, we will find that most CRCs, more than 80%, will have the APC loss of function mutations, and this will lead to constitutive and consistent activation of the Wnt beta-catenin pathway. The activated beta-catenin pathway will lead to the translocation of beta-catenin, and it will activate several oncogenes, in contrast to the inhibited Wnt pathway. If we go to the other and here there are several crosstalks in Wnt pathway. The MAP kinase pathway actually requires different KRAS and BRAF mutations and will lead to different effects like angiogenesis, like inflammatory response, and this will finally lead to tumor growth and even accelerates the process of metastasis. And in fact, this the kinases in general will have different parallel pathways that are activated. And that's why in order to treat those patients with those types of mutations, you have to use combined therapy to attack different pathways at the same time. And almost similar in the PI3K, AKT, mTOR pathway vulnerabilities, here we are talking about several pathways that have crosstalks among each other, leading to several hallmarks of the tumor, including the initiation of the tumor itself, the vascular stimulation and the angiogenesis, self-renewal, and the drug resistance development. Other pathways will lead to proliferation, invasion, and finally the growth and survival. So this sort of AKT or PI3K, the kinase capability will lead to several factors, including the epithelial mesenchymal transition that induces the colorectal cancer metastasis. The DNA damage response and replication stress is a quite important factor that is needed for survival of a cancer cell. And whether we are talking about defects in mismatch repair genes or the presence of replication stress in chromosomal instability high tumors, or targeting certain genes like ataxia-telangiectasia receptor gene or CHEK1 or PARP, all of those are very important pathways that are needed for DNA repair in a cancer cell that keep it surviving. So whether we are talking about germline mutation or some hereditary types of colorectal cancer or epigenetic silencing of certain genes, like I've explained in the early slides, the consequences are many because there will be accumulation of certain mutations with the insertion or deletion, then hypermutated phenotype and increased neoantigen formation that leads to more or higher level of proliferation of the cancer cell. Also increased sensitivity, but this is actually has the neoantigen load is a good determinant and a good indicator of response to immunotherapy. The increased sensitivity to PARP inhibitors, a particular class of medications that under specific contexts can have synthetic lethality with certain DNA mismatch repair gene defects or mutations, and finally the increased replication fork instability. So all of those consequences can be used to design the therapeutic regimen for those patients with defects in mismatch repair. For the metabolic reprogramming, there are certain dependency on certain amino acids. And of course, this is the cause why people are saying that intermittent fasting can be helpful for cancer patients. And it's actually a long controversial issue because we know that many, I mean, theoretically speaking, the amino acid deprivation or restriction can help those patients, can help actually the survival of the cancer cell in vitro, okay, in the cell culture. However, if we are talking about a patient who has specific nutritional status, some general health problems related to the cancer, it is really very critical to advise the patient to follow intermittent fasting at certain stages of the cancer. However, we have published this paper showing the different nutritional dependencies of certain cancer and how it is related to specific therapies. For example, the histidine supplementation together with methotrexate, or the serine restriction together with biguanides, or and all of those are actually proven in previous studies. This is just a review article published by a master's student, actually, whom I supervised a few years ago. We also have the epigenetic vulnerability. Another important approach to prime the cancer cells and sensitize it to therapy is to use the epigenetic modifiers like the histone deacetylators or the DNA methylation modifiers. And I'd like to refer you to an interesting chapter that I authored in this book together with two colleagues on histone modifications in cancer. The final one is the tumor microenvironment, and one of the most important factors that determines the response of the immune system to the cancer cells is the presence of the T lymphocytes in the surrounding microenvironment, the surrounding tissue, or the microenvironment, the tumor microenvironment. And that's why we can classify the tumor microenvironment into either hot microenvironment, which is studded with CD4 and CD8 T lymphocytes, in particular CD8, which are the cytotoxic T lymphocytes. And in contrast, there are some tumors that are they have cold tumor microenvironment, so the infiltration is not that huge. And the interesting idea is to turn the cold tumor microenvironment into a hot one. And in fact, we have I have like two publications, again with the PhD students and the postdoc, one on the role of autophagy inhibitors and how it can turn the tumor microenvironment from cold to hot, and whether this is effective in enhancing the effect of immunotherapy like anti-PD-1 or anti-PD-L1 in colorectal cancer. So you are trying to prime the tumor microenvironments in order to help the effect of the immunotherapy. So in short, we can summarize the therapeutic strategies of using such vulnerabilities into either using the combination therapy in order to attack different nodes on the same pathway, what we call the vertical inhibition, or sometimes you may attack several pathways in parallel. As an example, the synthetic lethality. And over the last 12 years, I've worked on different targets of colorectal cancer. And one of the interesting studies were done on the dual kinase DRCA1A, which is actually involved in several diseases like Down syndrome, diabetes, etc. And actually it has a contextual role, a dual role. Sometimes it works as a tumor suppressor gene, sometimes it works as an oncogene. And in fact, the DRCA1A as a dual kinase has an important role also in the DNA repair, like in the non-homologous end-joining repair, as well as the single-stranded anneal. And the studies done on the DRCA1A showed that it can be a good adjuvant to the medications that works on the G1 S phase arrest of the cell cycle. And we tried it actually in vitro on several medications like topotecan, cisplatin. Both are working as G1 S phase specific versus paclitaxel, which is a late G2M phase specific medication. And we found that the synergistic effect appeared clearly when the DRCA1A inhibitor is used with the G1 S phase specific medications. in vivo experiments also showed that the medication can decrease the tumor size, and I mean, using it as an adjuvant can decrease the tumor size as well as the tumor weight, and even it can decreases the metastasis in terms of the nodes that appears in the lung as metastatic lesions. In another study, we tried to exploit the autophagy, which is a biological process occurring in every cell, and its modulation when combined with IGF-1 receptor inhibition or silencing. We know that IGF-1 receptors are stimulated particularly in link to overweight and obesity. And using the autophagy inhibitors together with the IGF-1R silencing, we hypothesized that it can actually lead to suppression of the tumor proliferation. In fact, here we can see that the combination of autophagy knocking out by using CRISPR-Cas9, so those cells ATG5 minus minus or ATG 7 minus minus means that we have knocked out the autophagy related genes 5 and 7, here 5, here 7, and there is also silencing of IGF-1R by using the RNA inhibition, and if we can, or the inhibition of expression, and here we can show, actually you can see that there is suppression of the epithelial mesenchymal transition, which is the important step in tumor metastasis. Here we can see that E-cadherin is upregulated, whereas vimentin and snail is downregulated. I mean, the expression of the protein is downregulated. This is the Western blot, and this is actually the flow cytometry showing the effect on the HCT116, an important cell model that we are using in colorectal cancer. And the same can be shown here. We started to find out the focal adhesion molecules, again, which are important indicators of epithelial mesenchymal transition, whether paxillin or zixin. And here we can see clearly that the knocking out of autophagy together with the silencing of IGF-1R can lead to suppression of the ability of the cancer cells to metastasize. So we started with transcriptomic analysis. Actually, in this study, we started with the transcriptomic analysis that showed us that IGF-1 is one of the most upregulated genes, the most significantly upregulated genes, and also the autophagy genes are the very important ones that are upregulated. And then we tried to find out the effect of inhibiting both, which can be considered a sort of synthetic lethality. Furthermore, we did a proteomic analysis on those cell lines that are treated with IGF-1 receptor silencing, and we found also that the transcriptional factor CLEF8 can be a potential therapeutic target. This is a transcriptional factor, but interestingly, even the transcriptional factor can be used as a therapeutic target, and there are nowadays some medications that are suppressing the transcription factors, which was thought previously to be undruggable. Here we show that the wound healing, which is actually the indicator of the cell migration, has been the cell migration has been suppressed by using this sort of inhibition of the transcription factor. Here the same, we are showing that the focal adhesion molecules and also the invasion of the cell have been suppressed by using the CLEF8 inhibition. In our transcriptomic analysis on cells that are knocked out for ATG or autophagy related gene number 7, we could detect that the DNA recombination pathways are the most significantly upregulated pathways. And this led us to the hypothesis that those particular cell lines have more tumor antigens or neoantigens. So they are more likely to respond to immunotherapy. So we hypothesized this research on having those simple experiments, and we actually finished the in vitro experiments. We finished also the in vivo experiments. We didn't detect any significant changes in the tumor size when we combined the autophagy inhibitor, either the hydroxychloroquine or the the 3MA, the different autophagy inhibitors, together with the immunotherapy. However, we are trying to find out whether there are any changes at the molecular level. So we have done some immunohistochemistry, and we have done the flow cytometry for different markers in order to detect which types of immune cells have been attracted to the tumor when we use the autophagy inhibitors. I hope that we can come up with interesting findings, whether in favor or to disprove the role of autophagy inhibitors as adjuvant therapy for different immune checkpoint inhibitors. Patients. And of course, using several medications in order to overcome the adaptive resistance have been now the strategy for a very long time. And that's why it's very important to have some therapeutic biomarkers in order to monitor the evolution of the cancer even after using therapy. So you can change the regimen of the therapy in order to fit the patient and the development of the new mutation. It goes without saying that using high throughput experimental platforms together with the bioinformatics analysis, the advances in computational biology in particular, had led to discovery of new targets, especially for the CRISPR-based genome editing, in which you can explore the different effects like loss of function, gain of function, epigenetic modulation. Even it can lead to appropriate prediction of what happens if you target several pathways in the cancer cell. It gives you an idea about the effect of synthetic lethalities, of cancer dependency, of the different mechanisms of drug resistance. And of course, this can allow the scientists to capture the context-specific vulnerabilities by analyzing the tumor genotype, but also the signaling networks and the microenvironmental composition. And this bridges the gap between genomic alteration and actionable therapeutic targets. The aim is to reach as much as possible what we call precision medicine, in which each tumor can be addressed by certain regimen of combinational therapy. So it's quite logic to target overactive oncogenic pathway, whether we are talking about driver dependency, because many colorectal cancers are dependent on certain pathways. I mentioned very early in this talk about the beta-catenin, Wnt beta-catenin pathway addiction. Also, the precision medicine alignment. We know that certain mutations are present and we can target them with certain therapies. Even the development of new drug classes like the CRC mutation inhibitor or the transcriptional modulators, they are actually effective in targeting certain pathways. But the paradoxical concept is to enhance overactivated oncogenic signals. So instead of inhibiting it, you are going to overactivate it. So what is the rationale of overactivation of oncogenic signaling? The core concept is that cancer cells operate within a narrow fitness window of oncogenic signaling. So too little means loss of proliferation. And what happens if you enhance this pathway too much? This will lead to oncogenic stress. And the oncogenic stress here means that the cell will enter into senescence or apoptosis or mitotic catastrophe. And this is demonstrated across multiple oncogenes and pathways, and is sometimes referred to as oncogenic overload or signaling toxicity or oncogene-induced stress or lethality. Still, the concept is new. It needs further validation and verification. And the most important question, at which phase of tumor development can we use this sort of overactivation of oncogenic signaling? And finally, there are still translational challenges for developing the best treatment and the best regimen for colorectal cancer. This is related to the intratumoral heterogeneity and even the different subtypes and molecular characteristics of different tumors, plus the clonal evolution. The tumor is always in a dynamic status, developing new mutations for resisting a therapy or keeping its survival. So actually it is a complex disease. The aim is to have biomarkers that are properly validated so we can use them as guidance for the therapy and for choosing the appropriate therapy for each patient. There is limited predictive power of single genomic alterations, so we have to get a global picture of the tumor, usually by multi-omic analysis, which is of course very costly, time-consuming, and impractical in most cases. Bridging preclinical models and clinical relevance is very important, not to translate all what we find in the in vivo models to the human being. It's still more complex in the human being with different molecular subtypes, with different mutations. So it's not easy to have direct reflection of what we find in the in vivo models to the human being. So in conclusion, colorectal cancer is driven by context-dependent molecular vulnerabilities, and exploiting those vulnerabilities require systems-level understanding of the cancer, multi-omics analysis. Basic science remains central to the next wave of therapeutic innovation. I'd like to thank all people who helped me during the last, let us say, 12 years in doing such studies, and also to the funding agencies from which I received grants in order to support my research. And finally, I'd like to thank you very much for your great attention and best greetings from the University of Sharjah, the cultural city of Sharjah in the United Arab Emirates. Thank you very much.